my my presentation is the role of glycoproteins in cell recognition and adhesion. So glycoproteins are membrane proteins that contain a carbohydrate chain, and these glycoproteins can be considered as N-linked or O-linked. So cell signaling involves the release of a signaling molecule from one cell that binds to specific receptors and results in change in the cells. So an adhesion refers to the linking of cells to other cells or to the surface to form an anchorage. So as I mentioned before, we have both N-linked and O-linked glycoproteins. So in N-linked glycoproteins, the first sugar residue is linked to the amide nitrogen of asparagine in the protein and have a minimum of 5 sugar residues. And in the O-linked glycoproteins, the carbohydrate is attached to the OH group of serine, terethionine, hydroxyserine, or hydroxyproline, and there's usually 1 to 4 sugar residues. So shown over here is a picture of the various different glycoproteins. So cell adhesion molecules, also known as CAMs, these are glycoproteins. They're involved in adhesion, and you also have proteins like integrin, lectin, and so on that are involved in cell recognition. And as you can see, these glycoproteins are in the membrane. However, the carbohydrate is in the extracellular part. So one specific type of glycoprotein is the CD4 glycoprotein that is found on T cells, and this CD4 glycoprotein is a membrane protein, and it's found in T lymphocytes. And basically what it does is it is a receptor that is responsible for initiate T cell activation during immune response. And it interacts with class 2 antigen to regulate the immune response. In addition, the CD4 receptor is also famous for HIV because HIV binds to these CD4 receptors in order to gain access to T cells. So a particular disease that a malfunction in glycoprotein is involved in is cancer. So typically, calcium-dependent interaction among E-cadherin molecules are necessary for adhesion junctions in areas of epithelial cell-to-cell contact. However, malignant carcinoma cells are characterized by poor intracellular adhesion and increased cellular motility. So defects in the E-cadherin expression results in the interference in the integrity of the adherent junctions, and these are observed in the carcinoma cells. So when there is loss of E-cadherin mediated adhesion, this results in your benign cells being transformed into metastatic cells because they can now move around freely and they're not attached. So shown over here is a picture of your E-cadherins. So the primary research article, or the title of the primary research article that I found was N-cadherin regulates molecular organization of excitatory and inhibitory synaptic circuits in adult hippocampus in vivo. So from this research article, I found out that N-cadherin, which is a glycoprotein, is involved in the regulation of synaptic plasticity. So the main question of this research paper was whether the role of N-cadherins at mature synapse is similar to that at development stages. So what they did was ablated all cadherins from mice exactly after they were born, and they basically tracked the development of this mice throughout adulthood. And the main test that they used there was the Morris maze, where a platform is placed in the water, and the mice basically finds that platform. So in normal mice, as shown in A, after repeated trials, the mice easily finds the platform, but as shown in figure B, after repeated trials, the mice with ablated cadherins does not find the platform as seen by the many lines. or the many errors it has made. So what this shows is that the novel function of N-cadherin and beta-catenin complex, the maintenance of neural circuitry, is necessary to generate flexible and persistent cognitive and synaptic function, which leads to the formation of memory. So my question is, how are glycoproteins involved in cell recognition, and which part of their structure makes it ideal for such a function? Thank you.